Welcome back, folks. 23.1, second part of this section, thinking about chemical signals and receptors. So what I want to talk about here, two things: amplification of signals and also different types of signaling molecules. So amplification is all about how very small quantities of ligand are able to effectively elicit a robust response from the target cell. So at each step in the resulting cascade of events after reception of the signal, a signaling intermediate basically stimulates the production of many more molecules needed for this next step in the signal transduction. So this amplification process occurs after signal reception and during the transduction process. And so this multiplication effect that you can see here in this figure is called signal amplification. There's two things. One, it speeds up the transmission of the information into the cell. And the second thing, obviously, is it amplifies it. So it makes a weak signal very loud so that the cell can have a completely unambiguous response. So if we look at the example of how glycogen is broken down in liver cells in response to epinephrine, adrenaline, so what we can see is that when we have a binding of a single epinephrine or adrenaline molecule to a G protein coupled receptor, that's one molecule binding, one receptor being activated, we then see this transduction cascade involving the activation of the G protein, the activation of adenylyl cyclase, the turning on of the production of cyclic adenosine monophosphate, the activation of protein kinase A, the turning on of phosphorylase kinase, and then the activation of glycogen phosphorylase, and ultimately the response of the cell to glycogen phosphorylase is the conversion of glycogen to glucose 1-phosphate, which is going to go into glycolysis and be metabolized there. So you can see that we go from one molecule to 10 to the 8 molecules of glucose 1-phosphate being produced. And this can happen very, very quickly. And so the speed of this response is partly due to the size of these second messengers like ATP being converted to cyclic AMP. So cyclic AMP is small and it's diffusible. And so because of its size, it's going to diffuse very, very, very rapidly. So these second messengers like cyclic AMP and calcium that we'll talk about later, they're small, very highly diffusible, and that means they can activate these enzymes. And then these, of course, are enzymes, so they have turnovers. That means they can activate other enzymes, and they're not inactivated themselves. So that also leads to the amplification. Let's think a little bit about signals. So cells basically have a limited number of basic signaling pathways defined by the genome, which produces the proteome, and so we've got to be limited by the realm of the proteome. And so, as well as having a limited number of signaling pathways, there are a limited number of signaling molecules, ligands. So many ligands are hydrophilic. Small proteins, short peptides, individual amino acids in the case of some neurotransmitters, they're derivatives, and then nucleotides or nucleosides. And you might want to look up the difference between a nucleotide or nucleoside. So these hydrophilic ligands tend to bind to transmembrane receptors. So a hydrophilic ligand is not going to pass across the membrane, obviously. And so that means its information is received on the cell surface. And so we could have a hydrophilic ligand activating a gated ion channel. So that's a ligand gated ion channel. And so the ligand binds, the channel open, could close, but here it would be open, and ions will move down their gradient, and that will elicit a response. We could have a system where a ligand binds to a G protein coupled receptor, and that in turn turns on a number of enzymes which interfere with second messenger levels, and that elicits a response. We could have a protein kinase being activated, so the ligand binds to a receptor, and then we have a protein kinase activating a molecule such as RAS, and then we have a lot of protein kinases in the transduction cascades giving us a cellular response. We can also have hydrophobic ligands. So these are going to be small, sufficiently hydrophobic so they can pass across the cell membrane. There they can bind to a cytosolic receptor, and that receptor can enter the nucleus and elicit changes in gene expression. So these are a lot of steroid hormones and the retinoids. So those are the kind of, the big groups of signaling ligand, signaling molecules, and also the big groups of types of receptor that we can have. 3 cell surface that we've talked about, and then 1 cytosolic. And we'll get into these in more detail in the next few videos. So I'll wrap it up there. When we come back, we're going to start talking about G protein coupled receptors. I'll see you in a little bit.